So today I'm going to talk about sort of the history of what I do and why I do what I do, starting with the basic science. So I was trained as a neuroscientist from a fundamental perspective, looking at basic cellular mechanisms, and my career, and that was back in 1995 when Dr. Volek was already doing ketogenic diet research. I was actually studying metabolism in vitro cell cultures. And that led me over 10 years to looking at ketones as a neuroprotective strategy. And over the years, we validated the effect of nutritional ketosis and exogenous ketone supplementation in a variety of model systems. And that has more recently led to human research that I will fill you in on a little bit as much as I can. So before I begin, I always like to thank our sponsors because they really do make this research possible. It's not always easy to get funding for this kind of research, especially at the federal level. The Office of Navy Research has been sponsoring me for 12 years now, and the Department of Defense actually funded a lot of the equipment that I'll be showing you in my talk. NASA actually supports us logistically to do various missions. One I'll be talking about the NEMO mission. And NAVC recently funded a project at Duke University to look at the ketogenic diet in divers. So we have funding from various private industries. My university is supporting us with an SBIR to develop our company, Ketone Technologies. And we work with a variety of organizations, 501c3 foundations, that can also benefit from this research on nutritional ketosis. So my university, says that I need to really disclose that the information that I'm presenting today is not medical advice. I'll be presenting some information on new strategies to implement therapeutic ketosis that you may or may not have heard of. And this presentation includes data on ketone technologies that some of it is patented by the University of South Florida, some of it's commercialized, and royalties go back and actually support our research. So the primary focus of my research over the last 10 years or so, and for the next 3 years, because I just got funding, additional funding, was to understand and prevent what's known as central nervous system oxygen toxicity seizures. So these are a limitation for our Navy SEAL divers using a closed circuit rebreather. Oxygen toxicity seizures are also a limitation of hyperbaric oxygen therapy, which is used for 14 different FDA-approved applications, including things like decompression sickness and ischemic wounds. So I was part of a NASA mission called NASA Extreme Environment Mission Operations, and I think we have actually 2 NEMO aquanauts will be attending this event, which is pretty unusual. And in this mission, we actually maintain living in an extreme environment of saturation, of pressure. And at the end of that mission, the NASA Extreme Environment Mission Operations, NEMO, is a Mars or sort of a deep space analog that NASA does. There's 14 different analogs, and it's the only one that actually has astronauts participating in it. And prior to coming up to the surface, you breathe oxygen to decompress, to denitrogenate. And when you do that, you breathe 100% oxygen at about an hour time frame to decompress, and that's really pushing the limits of CNS oxygen toxicity. A Navy SEAL diver just at 50 feet of seawater can only stay there for about 10 minutes before they have the potential for oxygen toxicity seizures. And we were breathing 100% oxygen at 60 minutes in that environment. So in that particular mission, I actually stayed in a state of nutritional ketosis. So we use a variety of tools really to understand fundamentally, mechanistically, and from a physiological perspective, what these extreme environments do. Elevated oxygen, elevated nitrogen, and also elevated CO2. We're becoming more interested in the effects of hypercapnia, elevated CO2, in the context of elevated oxygen levels. And we use a wide variety of tools, including things like atomic force microscopy, scan and confocal laser microscopy, electrophysiology, and radiotelemetry. And we've adapted these tools for use inside of hyperbaric chambers, like the one shown here, where we have atomic force microscopy and confocal microscopy. And this allows us to look at things really at the basic fundamental level, to where we can look at the cell membrane, the viscoelastic properties of the cell membrane. We can quantify, for example, oxidative stress as a function of oxygen and how that changes lipid aggregates or the actual composition or surface of the membrane. And with confocal, we can look at the mitochondria. We can optically section the cell and look at mitochondrial reactive oxygen species production as a function of oxygen over time. So on the other end of the spectrum, we do whole animal research using radiotelemetry, where we do EEG measurements, ECG, and diaphragmatic EMG to look at respiration. And using this equipment, it kind of allows us to vet out various neuroprotective strategies to find out what works from like a top-down approach. And then we can go back, at this point in time, go back mechanistically to determine how particular ketogenic agents are functioning from a mechanistic perspective. So we have the ability to do EEG studies, so we can detect in very high resolution the occurrence of a seizure almost before it happens, because there's some cardiorespiratory biomarkers that we think can predict an impending seizure that might be useful. And now we're actually looking at and vetting out different detection strategies and mitigation strategies. So in 1995, my primary focus, and kind of still is today, is neuroscience. And I was investigating the effects of low pressure oxygen and high pressure oxygen using in vitro cell culture preparations and also brain slice preparations, as shown here. And it was really in an effort to understand fundamentally, as my PhD work transitioned to my postdoctoral fellowship work, was to really understand fundamentally why cells overproduce oxygen-free radicals and why this may be contributing to metabolic dysregulation that contributes to these tonic-clonic seizures that are a limitation of Navy SEAL diving. So to do this, we use a variety of preparations, and one was the hippocampal brain slice preparation, so where we essentially can cut a living brain into a tissue slice, like a pizza bread, and that has the intact cytoarchitecture, so we can measure the electrical activity in the brain under different parameters. And in this study, we measured the effect of different levels of oxygen from 20, 40 to 60 to 95% oxygen. And in the middle picture is a snapshot of the hippocampal brain slice preparation of the CA1 region. And the fluorescence that you see there is a dye, we use dihydroethidium, and that detects the formation of superoxide anion, which is a precursor free radical for other reactive intermediates that we think disrupt neuronal function and start to disrupt also glucose metabolism. So this preparation also allows us to look at seizures. So in this image here, we can detect the orthodromic population spike, which are all these neurons firing together after being stimulated. So we stimulate one time, and that the amplitude of this orthodromic population spike measured under hyperbaric oxygen conditions, you could see relative to control conditions, there's a greater number of neurons that are hyperexcitable and firing. When we increase the level of oxygen, not only do we get a big amplitude in the orthodromic population spike, we get these secondary spikes that occur from a single stimulus. So these secondary spikes are analogous to a seizure in a slice. So, and as you may know, the ketogenic diet really evolved as an anti-seizure strategy. So at this time, I started becoming more interested in the ability to administer a ketogenic therapy to prevent oxygen toxicity seizures. And in my early work, back in the mid-90s, I was actually interested in a molecule called alpha-L-polylactate. So I was mostly interested in lactate, but it didn't really have an effect in our preparation. So as a continuation of this work, going from cellular studies to animal studies, we started doing these telemetry experiments to vet out, different agents that could potentially mitigate, be a countermeasure to oxygen toxicity seizures. And as that work evolved, and I'll get to more of this later, we started collaborating with organizations or facilities like Duke University, where they have a hyperbaric chamber, and they can do a wide variety of experiments to also fundamentally understand what is contributing to oxygen toxicity seizures. And they study things like nitrogen narcosis and also decompression sickness. So in this experiment, or in this setup here, they don't actually push the divers to a seizure, but there are a variety of physiological biomarkers, cardiorespiratory biomarkers, that can actually detect an impending seizure before it happens. And we're doing a study right now. It's double-blind, it's placebo-controlled study looking at this effects of nutritional ketosis. So, and also we, as part of the NASA Extreme Environment Mission Operations, we did a variety of cognitive function tests. Here's me using an iPad underwater at about 3 atmospheres, breathing hyperbaric air, and doing things like looking at reaction time, looking at decision making. So under these extreme environments, we're at the point now where we're collecting data. We want to collect data from at a minimum of 2 or 3 missions, and then that data sort of becomes the baseline data to compare a ketogenic intervention. Although in this particular mission, I actually stayed ketogenic because that was my baseline. So there are a variety of things that we can do to prevent oxygen toxicity seizures. Not all of them are feasible. For example, according to the dive tables, the Navy dive tables, if we adhere to strict exposure time, that will decrease our chances for oxygen toxicity. So at 30 feet of seawater, our length of exposure is roughly 80 minutes, but at just 50 feet of seawater, our length of exposure is just 10 minutes. And after 10 minutes, we have the potential for oxygen toxicity seizures. And this varies considerably between different people, and it can also vary considerably between the same person. If one person, the same person, is sleep deprived or on high levels of decongestants like ephedrine, pseudoephedrine, that can actually increase our predisposition for oxygen toxicity seizures. So there's a wide variety of things that could augment or initiate oxygen toxicity seizures. During my initial studies, I became mostly interested in antioxidant compounds, things that can prevent the formation of reactive oxygen species, which I thought was disrupting normal brain metabolism and the neuropharmacology of the brain. So antioxidants tend to work very well in vitro cell cultures and in brain slice preparations, but they don't work very well in vivo, in whole animal studies. So the go-to drug really for oxygen toxicity as it pertains to sort of like a disabled sub-scenario where you need to get the guys denitrogenated and up to the surface would be anticonvulsant drugs like vigabatrin and other drugs at very high concentrations or doses that could impair warfighter performance and capabilities. So I became increasingly interested in alternative strategies for this, and one of the things that caught my attention was the observation, and this was documented by 2 independent labs, that fasting could actually delay the latency to seizure up to 250%. And this was above and beyond any anticonvulsant drugs that I saw. There was a few experimental compounds that reached that level. But this idea of I didn't know how it worked at the time because and fasting didn't seem very feasible, but mechanistically I became interested in it. So if I can get this to advance. So the question was, how does fasting change brain energy metabolism? So the work that was referenced before by Dr. George Cahill really inspired me to go in this direction. And when I first read this study, I actually had to call him. He was alive at the time. This is prior to 2008 or 9. And I asked him for some of the details of this study, which was pretty remarkable. And there was some other things that he did, maybe some of it not published. But it was something that could probably not be reproduced today, where they fasted subjects for 40 days. And prior to 1967, when this was published, most people were under the impression that glucose was the exclusive fuel for the brain. There was some people looking at lactate and amino acids, but as a primary brain fuel, it was thought that largely 100% of your energy metabolism needed to be derived from glucose. And that's true in a fed state. If you're eating a normal diet, roughly 100% of your brain energy metabolism is coming from glucose. But after about a week to 10 days, you could see that the level of beta-hydroxybutyrate and other ketones like acetoacetate are also elevated. After about 10 days, the ketone body beta-hydroxybutyrate becomes the primary substrate for brain energy metabolism. And he looked at the AB difference in these subjects over the 40-minute time point. And you could see glucose does not go into severe hypoglycemic ranges, right? Because we have very powerful homeostatic mechanisms that maintain our blood glucose. There's gluconeogenesis from gluconeogenic amino acids. The glycerol backbone of triglycerides becomes a source for glucose over time. So in an extension of the study that was not actually published in the original work, but is found elsewhere, they injected these fasting subjects with 20 IUs of insulin, which if you're on a normal diet and your glucose is suppressed with an insulin administration down to hypoglycemic levels, severe hypoglycemic levels. So I teach the medical students, and we teach that if you're under 2 millimolar, you typically go into a coma, and you're not and at 1, you're basically approaching death. So these subjects experience severe hypoglycemia in a fasting state with insulin, and so just as reference, that would be around down here, the level of glucose, and they were asymptomatic for hypoglycemia. So after reading this and talking with Dr. Cahill, I realized that this had profound implications for safety and also for performance. We know when your blood glucose dips, your cognitive and physical performance also dips. So I became increasingly fascinated with how to employ this approach in my studies. So historically, there are really about 2 ways that we can sort of induce therapeutic ketosis. There's starvation, no one wants to do that. There's calorie restriction, and you can only maintain that over a certain period of time. And there's things like intermittent fasting too that have been more popular recently. There's also the ketogenic diet, and there's a lot of discussion as to what is the optimal ketogenic diet to maximize the therapeutic effects of nutritional ketosis. So the diet that we pretty much employ, that I use myself and that we use in our studies, is about 20% protein and 70% fat. And a lot of people ask me, it's like, well, will this kick me out of ketosis if my macros are this or that? And my response is just measure. Use the commercially available tool to determine if your particular. So a ketogenic diet is defined, it's the only diet that's defined by an objective biomarker that we can measure, right? Our urine or blood ketones. And that makes it kind of a unique thing from a scientific perspective. So the ketogenic diet is maybe not difficult for you all to adhere to, but for the large majority of the population, it is difficult to sustain day in and day out. And you need to consistently decrease, liver glycogen and maintain that decrease in liver glycogen and suppression of insulin signaling to drive hepatic gluconeogenesis. A small carbohydrate meal can quickly kick you out of nutritional ketosis, and if you are trying to mitigate or prevent seizures, that can trigger a seizure. So more recently, over the last 10 years or so, there are various things that can be employed that can induce nutritional ketosis and circumvent the dietary restriction that's typically associated with getting into therapeutic ketosis. And we know that ketones are viewed as an alternative form of energy. So when it comes to, the most potent ketogenic agents, I would say ketone esters can put your ketone levels pretty much wherever you want them to be. And that could be different depending on whether you're using it for seizures, performance enhancement, or whatever your approach would be. So we know that ketones are an alternative fuel for the brain and the heart and other tissues like the muscles. And more recently, over the last 5 to 10 years, we understand that they're very powerful signaling molecules from the perspective of a neuroscientist. This is one of the graphs that I frequently show and kind of walk through it. In the interest of time, I'm not going to walk you through this, but we are interested in things like adenosine is functioning. Adenosine goes way up with the ketogenic diet in tissues and in the blood. And also the GABA to glutamate ratio is increased. So glutamate's a hyper-excitatory neurotransmitter, and GABA is more of an inhibitory neurotransmitter. And glutamic acid decarboxylase is an enzyme that converts that makes that conversion. So the ketogenic diet and more recently exogenous ketones have been shown to increase the enzymatic conversion of glutamate to GABA. So that's an area that we're focusing on now. And we heard recently that in the last talks that beta-hydroxybutyrate is a histone deacetylase inhibitor. So through acetylation of proteins, that can have tremendous epigenetic effects. Now we're studying a disorder called Kabuki syndrome. And we think there was some work done at Johns Hopkins, and we're transferring the Kabuki syndrome mouse, and their observations demonstrated that the epigenetic therapy of beta-hydroxybutyrate employed with a diet or a ketone supplement actually saved the Kabuki phenotype in regards to it prevented the neuronal degeneration in the hippocampus. So we want to validate sort of some of the exogenous ketones that we're studying in the lab right now. We know that the NLRP3 inflammasome is suppressed by beta-hydroxybutyrate. So that has wide-ranging implications on a wide variety of diseases. And there's a number of ketogenic or ketone receptors to beta-hydroxybutyrate, and more recently acetoacetate has been shown to be a receptor to that. So as you can imagine, there is an emerging number of applications to therapeutic or nutritional ketosis. And nutritional ketosis can be employed in different ways through dietary means and through exogenous ketone supplements in the form of ketone salts, ketone esters, or medium-chain triglycerides. And when I first started studying the ketogenic diet in 2008 or 2008, I believe, the only therapy that it was really sort of validated for was pediatric epilepsy. And over the last 10 years, you can just look at PubMed and see the explosion of research on nutritional ketosis and ketones. So I put two categories here of strong evidence and emerging evidence. And we know that we've had historic data that low-carb diets, ketogenic diets can promote weight loss and the maintenance of that weight loss. So, and I think that's the key factor, right? It's pretty easy to lose weight, but it's more difficult to maintain that weight loss. And at least the people here, are under the impression through their research that you can maintain that weight loss. Type 2 diabetes there wasn't a whole lot of research on this about 10 years ago. Over the last 10 years, through a number of studies, some of them funded by Virta Health, very compelling data that type 2 diabetes, and I know we'll hear more about that in the upcoming talks, people with type 2 diabetes are very responsive to low carbohydrate diets and ketogenic diets. And inborn errors of metabolism, including pyruvate dehydrogenase deficiency complex, the ketogenic diet's actually the front line of therapy for that. For epilepsy, it's sort of the fallback therapy for drug refractory or drug resistant epilepsy. It's not to the point now where it's frontline therapy, but for things like pyruvate dehydrogenase deficiency syndrome, it is the frontline therapy. So for type 1 diabetes, I know my student Andrew Kutnick may argue he has a poster on the use of carbohydrate-restricted diet for type 1 diabetes that you might want to go to. There's emerging evidence, and maybe strong evidence, that type 1 diabetes can be managed There's far less variability in your glucose and a huge decrease in the dependence of insulin to manage type 1 diabetes with nutritional ketosis. Non-alcoholic fatty liver disease, things like polycystic ovary syndrome, brain tumors and cancer. My research associate, Dr. Angela Poff, will be presenting on that tomorrow. There's a number of applications, and when it comes to neurological applications, we study glucose transporter type 1 deficiency syndrome. Again, this is a neurological disorder that is highly responsive to the ketogenic diet. If a kid has glucose transporter type 1 deficiency syndrome, they really need to be on a ketogenic diet for quality of life and to just maintain, to be able to function. So it's a very powerful approach. And in some cases, it's very difficult for that child to employ the ketogenic diet and to stay with it. So that's why some of the things that we're doing, developing technologies that can induce therapeutic ketosis and circumvent that dietary restriction that not all people can follow, is important for that community. Dravet syndrome, Lennox-Gastaut syndrome, of course, epilepsy. Adult epilepsy, the ketogenic diet was really only sort of embraced for adult epilepsy not until about 2008 or 10, and they use a modified ketogenic diet approach. So when it comes to emerging evidence and applications, we're going to hear about Alzheimer's disease, autism. Angelman syndrome is something that we study. We study the Angelman syndrome mouse model and have moved to actually a clinical trial for Angelman syndrome right now using ketogenic supplements. Kabuki syndrome is another project that we are just starting up right now. And because lack of space, I put anxiety on there, but anxiety is also something that adults with Kabuki syndrome experience anxiety. And we have a number of publications to show that anxiety behavior goes down in a state of nutritional ketosis. So that's another emerging application. I don't really have time to talk about it today, but that may be due to a lowering of glutamate and an elevation of GABA, which is more of an inhibitory brain stabilizing hormone. Neurotrauma, traumatic brain injury. With traumatic brain injury, you have glucose impairment or impairment of glucose utilization that can be detected by an FDG PET scan, for example. And about 80 to 90% of people with penetrating traumatic brain injury have a seizure. So it makes sense that if you've had a concussion, and if our warfighters are coming home with traumatic brain injury, we think it's very important for them to employ nutritional ketosis to, at the very least, to prevent the seizures that are very common with penetrating traumatic brain injury. So the last two things I want to focus on, and they relate more from an operational standpoint from the warfighter or maybe even the astronaut. We're going to look at anesthesia resistance and the employment of nutritional ketosis or therapeutic ketosis for oxygen toxicity seizures. And this is work that we did it was published back in 2013. We started these studies in 2009 or 10, and it took a while to sort of vet out and find the ketogenic agent that was most neuroprotective in this regard. And we tested a number of different things when we did this. But under normal baseline conditions, if we expose a rat to 5 atmospheres of oxygen, which is equivalent to 132 feet of seawater, that produces these tonic-clonic seizures within about 5 to 10 minutes. So, and the therapeutic range of ketosis that we need to get to is anywhere between 2 to 5 millimolar, at least with a ketone ester. And there needs to be an elevation of not only beta-hydroxybutyrate, but also an elevation of acetoacetate. So we used agents that elevate beta-hydroxybutyrate exclusively, and we did not have anti-seizure effects of these agents. So we used a ketone ester that elevates beta-hydroxybutyrate more or less in a 1-to-1 ratio. So after about 10 minutes, rodent models, usually have a seizure, mice and rats, in about 5 to 10 minutes at 5 atmospheres of oxygen. If they are acutely given a ketone ester, meaning they're eating a high carbohydrate standard rodent chow, and we administer a ketone ester orally, and then we dive the rat in the hyperbaric chamber, we typically see resistance up to 60 minutes. So in our study that we published, there was a 575% increase in the latency to seizure. And that's above and beyond any anticonvulsant that I know of. Vigabatrin at very high levels gives you about 400% anticonvulsant effect. So we're giving a metabolic substrate that has a greater anti-seizure neuroprotective effect than any anticonvulsant, experimental, or FDA approved that we know of. So this was sort of our first study that we did, and now we're doing a number of studies looking at a half dozen different agents and combinations of things, and looking at specifically CNS oxygen toxicity. More recently, we made the observation that in studying the glucose transporter deficiency mice, these mice go down very fast with anesthesia because their brains are they're kind of on the edge, right? Because they're very hypoglycemic. The CSF of glucose transporter deficiency mice is about 2, maybe 2.5 millimolar of glucose. So their brains are kind of starved for energy as is. And we noticed that they go down very fast with anesthesia, but if they're in a state of nutritional ketosis with either the ketogenic diet, a ketone ester, or a ketone salt, that the latency to their anesthetic induction, which is the point where they fall over and they're immobile, was greatly increased. So my wife was doing a lot of these experiments, and members of our lab really observed that these mice were much more resilient to anesthesia. So it became, this weird observation actually became a study in and of itself in a recent publication. So we validated this. It made sense in the glucose transporter deficiency mice, but we also did studies using normal, healthy Sprague-Dawley rats, and we observed that a ketogenic diet and to a lesser extent ketone supplements delayed the anesthetic effect of isoflurane. So, which is an anesthesia that we use very commonly in our rodent models, and also a rat model, the WAGRI rat model that our collaborator uses in Budapest, is a rat model for absence seizures. And we observed also a pretty profound delay in the latency of seizure with the ketone ester. So one of the big projects we're sort of spearheading right now is to look at the effects of nitrogen narcosis and CO2 narcosis. So if you work with animals, CO2 anesthesia or CO2 narcosis is what you use to put the animal down before you kind of study him. And also nitrogen narcosis is experienced by divers. If you're breathing air, for example, with standard scuba, and you go down to 150 feet, you will experience what's called the martini effect. So you will basically be drunk from nitrogen. So the anesthetic potency of a gas is proportional to its lipid solubility, and halothane or isoflurane is very lipid soluble, so it has a very potent anesthetic effect. Nitrogen's not very lipid soluble, but at high pressure, it becomes a pretty potent narcotic agent. And CO2 is also a very potent narcotic agent. So this is relevant to an operational setting, also because CO2 is a big problem. It's a big operational problem, we could say. So outdoor, right now, we're breathing anywhere between, depending on if you're inside a car without the ventilation on, or you have a motorcycle helmet on, the levels can get higher, but typically it's about 350 to 400 parts per million, or 450 parts per million of CO2. So the OSHA limits is about 1,000 parts per minute. So that they determine that's sort of like the safety limit. And then if you're above that, then you start to experience general drowsiness. And if you're above, say, 2,500 parts per million, you start to get adverse health effects. So I've been involved in a number of animal studies where we look at things like the immune system, we look at the brain activity, we look at the gut, the tight junctions of the gut. If you're above 3,000 parts per million, it starts to break the tight junctions of the gut and makes you have a leaky gut. So these are some of the things that sort of caught my interest to put more attention into CO2. And also if I can get this slide to advance. So in the context of an operational setting, World War II subs really approached levels, in some cases, up to 4%. and that could be 10 or 20,000 parts per million. So they were exposed. Today's submarines are actually somewhere around 3,000 parts per million, and they're trying to bring that down. In the case of a disabled submarine, the levels of CO2 can get very high, and that can have a very potent narcotic and even deadly effects. So many of the scenarios where people die in these operational environments, whether it's a spaceship, Apollo 13, they had the CO2 problem, or in a submarine setting, CO2 really is one of the biggest problems. So it's really important to understand it and also to mitigate it. And so on the International Space Station, they experience a level of CO2 that's about 10 to 20 times higher than the level of CO2 we're breathing right now. And that's also very similar to the NASA analog that I was involved in, the NEMO mission. So during the NEMO mission, I periodically took pictures of the CO2 monitoring system. And at one point, it was 1.27 and actually got up to 2% oxygen, which was about 20,000 parts per million. So this is way above the safety levels. And on ISS, they're living there for months. But in this space analog, we were there for 10 days. And I measured in myself, a lot of urine, blood, and saliva measurements to look at how this was affecting my physiology. And we also measured things like sleep and heart rate variability that we still have to analyze. So we think that this is an important problem. And it's important to employ, we believe, studying it first at a basic science level and also a whole animal model level to determine practical implementation of countermeasures and also monitoring. So when it comes to the ketogenic diet, here's an example of a meal that I ate, on the NEMO mission. Actually, this meal, I would either put coconut oil or MCT oil in a can of chicken, and I had macadamia nuts. So the idea, you can't really get a lot of fresh fruits or vegetables in this environment. So you have to your strategy needs to be pretty much canned food. And this is a meal that I had last week, and it was a pizza that's on the market now. I see an explosion of different foods that are low-carb foods or even ketogenic foods, and a salad and cookies that one of my students made that are ketogenic. So a variety of new technologies are helping us to prepare foods that are thought of as comfort foods, but they can have the macronutrient ratios or even ingredients in them that make them ketogenic. So on the NEMO mission, I was actually, after an EVA where we go outside the habitat and work in the water, I became mildly hypothermic. And I experienced that because I was shaking when I got back. We were out in the water for about 6 hours. And my blood glucose measured when I came in 2.3, and my ketone levels were 6.9. So my ketones were then at that point in time, by far the primary fuel for my brain and functioning. And during that time, I actually took, a number of cognitive tests outside in the habitat, looking at reaction time and things like that. And there was no impairment, at that level. And eating on a day-to-day basis, what I call a modified ketogenic diet, which was described by Dr. Finney and Dr. Bullock, it brings me to, at the end of the day, a level of glucose of 3.3 and 3.3 ketone levels, which gives you a glucose ketone index of 1.0. And we use that glucose ketone index sort of therapeutically because we know a wide variety of diseases will respond to that from an anti-seizure perspective and also from an anti-cancer perspective. So there's a number of monitoring technologies that are out there, from urine to breath to blood, and also future devices that are being developed that can measure, continuously, like the glucose measurement, like the Dexcom, potentially beta-hydroxybutyrate, acetoacetate, and lactate. I'm communicating with a few companies who think this technology will be available in the next 2 or 3 years. And that could be very useful if you're using the ketogenic diet therapeutically or also from a performance standpoint. So in summary, and I want to kind of go through each one of these very fast and to determine or just mention the key questions that each of these things each of these projects are looking at. So the recent grant that I got funded through Office of Navy Research is focused on optimizing ketogenic metabolic therapy, looking at various ketone technologies, whether it be an ester or a salt or even medium-chain triglycerides, to mitigate oxygen toxicity seizures. And we're also looking at various predictors of oxygen toxicity seizures. We're looking at CNS oxygen toxicity in the context of hypercapnia. We know high levels of CO2 increase brain blood flow and oxidative stress, so we want to understand that. There's a mouse study we're doing right now on Kabuki syndrome, and we're looking at the epigenetic effects of beta-hydroxybutyrate. And also, more recently, we started the human studies, which include the study at Duke University, funded by NAVSEA, to look at the effects of the ketogenic diet on early biomarkers of oxygen toxicity seizures. And we're collecting a lot of data, 5 IRBs worth of data, on the NASA NEMO missions, including 22, which I was on. 23 is coming up, and next month it's going to be C-Lab or C-Test 5 mission. And each of those missions will have 6 people, and as we collect data, we'll understand what this extreme environment does to a wide variety of biomarkers, and potentially how a ketogenic intervention can help enhance our safety and resilience in those environments. So I want to thank you for your attention, and I'll be here for questions after. And although I don't really have time, there's one last slide that I wanted to quickly show because I get this a lot. How do exogenous ketones impact the glucose levels? So we've shown in mice, rats, and humans that acute administration of exogenous ketones in the form of ketone salts, ketone esters, and even medium-chain triglycerides lower blood glucose profoundly. And I think that may be, I didn't get to talk about it, but that may be one of the most important sort of therapeutic effects of exogenous ketones. Thank you.